In this video, I'll be taking you through the steps of doing an antibody validation. So I'm going to go ahead and open up an existing project, and from here I'm going to go to Create New Experiment, Quantitative Western Blot, Antibody Validation. And so we've already imported the images into the image gallery, so I'm going to click on that, scroll down to the image that I need, this one right here. And I'm going to select the 700 channel first, and then it's going to give you automatically the choice of also adding the 800 channel as well. So I'm going to go ahead and click the add for that. If you have a third channel that you would like to add, you can certainly do that here as well. So this is going to be my tubulin COX-4 antibody validation. If you want to, you can add a description here at this point. Go ahead and click on Next. And here in the 800 channel, I have the Odyssey loading indicator. So I'm going to click that. And you can add all the information here that you need. This is Odyssey loading indicator, 28 kilodaltons. You can add all of the vendor information. The product number, lot number, dilution, incubation times, species, diluent, immunogen, the product page link if you need to as well. And for the secondary antibody, this has no secondary body that was used, so I'm going to go ahead and click on that and say OK. And then for the 700 channel, I have tubulin, and I'm going to add the information here as well. And then I'm going to fill out all of that information. For the secondary antibody, I have IRDI 680 RD, and then you can fill all of that out as well. And so this will all be part of the report at the end, and also all of this will be added to the antibody validation database. So then we also have on here, we have COX-4, and again, you fill out that information. Add all of that right here and click OK. Now, it's a very good idea to fill out that information as fully as you can, because that's very useful later on when you wrap up your project. Go ahead and click Next, and we're going to draw boundaries around the outside here of all of my lanes. And so what you're looking for is that the line is going essentially through the middle of all of the lanes on here, and so that those all look good. If you need to do manual adjustments, you can click on Manual and then set up the number of lanes, and from there you can adjust the location or angle of the lanes if need be. But I'm going to go ahead and stick with the auto for this one. Click Next. And then you're going to set up your lanes. So I have lane 1 here. This is my molecular weight marker. Lanes 2, 3, 4, and 5 are all samples. Lane 6, again, is a molecular weight marker. And then also the same with lane 11. And then if you want to, you can add any kind of information as far as the amount of extract that was used, concentration of the drug target that you used, or dosing or treatments, whatever you want with there. If none of those options are valid for your experiment, you can add a new label for that, just give it a new name. Same goes with the unit labels. All of these options are here, including percentage, but if none of those fit, you can add a new unit label there as well. And so for the molecular weight markers, go back here and get that set correctly. For the molecular weight markers for the 700 channel, I'm going to choose the Chameleon Duo 700. And if you have a different marker, because if we look at the information here, if these weights on here do not match up with the weights that you have actually loaded here on the lanes, if you've used a marker from a different company, or if the bands, or if the let's say the top one up here did not get resolved, you can remove that if need be, and then save that as a marker that you can use at some later point. So I'm going to go ahead and click Add, for all of these. You notice that I don't have to go top to bottom, because as I move along, it does adjust those assigned weights accordingly. Then I click Done. Then for the 800 channel, I choose the Chameleon Duo 800. Again, I click Add, and I go through the same process of clicking on the bands for weight determination. Click Done, and then click Next. So for the quantification, so here this one is selected, the 800 Odyssey loading indicator. I'm going to click on Find Bands, move this to go through as many of the bands as possible. And at this point, if I need to make any adjustments to the size or location of these band markers, I can do that right there. And then I want to go over to the 700 channel to do the same process. That one for the tubulin, I'm going to click on the Cox 4, click Find Bands. Again, try to cut through as many of those as possible. Again, if you need to make any adjustments to those band markers, you can do that there. And so then here under the Review and Reports, this will give you quantification of each one of the bands that you found on here. It'll give you the molecular weights, signals noise ratios. It'll give you all of the antibody information here that you entered earlier. Same holds true for the tubulin one that was done, and then also the Cox 4. So from all of this information, you can make a judgment on how good of a primary antibody it is, along with the secondary antibody that you used. And if you want to export any of the data themselves, you can click on the analysis table, export table to a data file, and it will create an Excel file for you. The same is true for the images. If you click on this export button, you can do a 600 dpi TIFF or 600 or 300 dpi PNG. You can make adjustments, or you can hide the lane lines if you need to as well. Your other options from this page are you can create a PDF report, which will include all of the images and data that are on this report right here. It just sends it out to a standard PDF file. You can export it as an experiment file, which allows you to transfer it to another computer and either continue or modify any kind of analysis that needs to be done. Thank you.